Good afternoon. My name is Mike Ward, and as Brian said, I'm the head of content at Informa Pharma Intelligence. That is a sister publication or sister company of bioprocessing, and I'd like you to welcome this discussion, which is sponsored by LabTwin. We're all aware that healthcare has undergone significant transformation over the past several years, moving from an antiquated paper-based record system to more efficient and integrated systems that often incorporate physician, patient, and patient-generated health data. This explosion of data means that there is a strong need and desire for systems that not only help and extract and organize this information, but will also analyze and even provide insights and recommendations on how best to utilize that data. Artificial intelligence, which is a phrase that is much bandied around, is rapidly being adopted across many different situations and use cases within the healthcare. In this context, artificial intelligence is an information system that is inspired by a biological system designed to give computers human-like abilities of hearing, seeing, reasoning, and learning. It's an umbrella term that obviously involves multiple technologies. In healthcare, AI is been largely implemented as a tool to move efficiently and accurately to remove or review data and uncover patterns in that data that can be used to improve the analyses, uncover inefficiencies, and streamline care in both clinical and operational activities. However, what we're going to discuss now is how AI also has the potential to be a powerful tool in the lab setting. That's the focus of today's session. What we're going to do is determine which research challenges AI can be used to resolve, and in particular highlight one solution which has been developed by our sponsor LabTwin, who is the sponsor of this discussion. And we'd like to open out I'd like to open out an invitation to the audience to participate in any questions that you have of the panelists or comment on anything that you hear. So first I'm going to ask each of the panelists to take a minute or so to introduce themselves, their sort of job title, their company, and also their sort of interest in this space. So
I'll start off. So my name is Ron Alpha. I'm SVP of Translational Discovery at a company called Recursion Pharma. So Recursion is a drug discovery company that uses a combination of machine learning on images with rich biological data sets that we generate in-house to do drug discovery at scale and in an accelerated way across many different indications. So a lot of the work that we do at Recursion involves using a combination of machine learning and biology to accelerate understanding of biology, but also involves building models that can help predict preclinical toxicity parameters very early in the process so that we can increase the efficiency and decrease the failure rate of drugs we develop.
Hi, I'm Krishnan Nandabalan, CEO of Manya AI Corporation. Manya AI Corporation is a company that uses machine learning and artificial intelligence in drug discovery and development. We are actually using machine learning to connect mechanisms of actions with pathophysiologies so that we can optimize the selection of disease indications for either first-in-class drugs or best-in-class drugs. And we primarily do this in collaboration with our collaborators, which are pharma or biopharma companies. And so the kind of data we deal with is not just one type of data. It can be omics data, it can be clinical trial data, and we also look at a lot of published data as well. So we build models to predict the efficacy or safety issues of a particular mechanism in the context of a disease. That's primarily what we do.
I'm CEO and co-founder of LabTwin, GmbH. Can you guys hear okay? Yeah? Okay. LabTwin is a voice first, but very much multimodal digital lab assistant for scientists and technicians in the lab. We use NLP and other machine learning technologies to enable much more connected, agile, and hands-free documentation and access to information in the lab from anywhere in the lab just by speaking to our solution. Our key goal is to actually make scientists or enable scientists to reproduce experiments completely worries-free and make sure that the documentation that they have to do happens completely autonomously, without actually scientists have to think or take time away from the experiment to document things afterwards.
Good afternoon. I'm John Piccone. I'm a partner at CES Associates. We're a global consultancy focused on analytics in life sciences, and I lead our discovery and translational medicine service line. Prior to ZS, I was at Watson Health for 8 years. I led the life sciences product portfolio, so AI products for drug discovery, real-world evidence, clinical trials analytics, and patient safety. We focus at ZS in our discovery and translational practice area primarily on aggregating and ingesting enterprise R&D data for cross-study analysis and providing analytics and analytical platforms as a managed service.
Thank you. I'm Seth Letterman from Tonic Pharmaceuticals, and we are a drug development company, a NASDAQ-listed drug development company. Our lead program is in phase 3 development, but our programs go all the way down to discovery chemistry. So we manage a lot of different kinds of data, and this is an exciting panel to talk about the complexities of all the data that companies like Tonic manages.
Great, so thanks very much. So in all the years that I've been covering the biopharmaceutical industry, so that's 35 years, one of the biggest challenges that I see that still remains unresolved is the transition of promising early stage science into meaningful clinical outcomes. In particular, different teams often find it difficult to reproduce results that have been achieved by others. So my first question is, one, I'm actually being fair in that observation, and how much of a problem is reproducibility, and what is being done to tackle it? So Seth, if we could start with you, that translational problem.
Sure. Well, before I went into industry, I was at Columbia University as a professor, had a 25-year career at Columbia, and I'm aware about concerns about reproducibility. A lot of the reproducibility issues that I'm aware of have to do more with university science, I think, than industry science. I think that the pressures to publish, to get promoted, to get grants and whatnot is a huge incentive for people to put out things a little early. And it's frustrating for companies, particularly companies in licensing technology from universities. Many times it's essential that you reproduce the systems you have. In a lot of cases, I think it's just that people are in a rush, but I think there are problems ultimately in the expense and the time that it takes industry, and it's one of the obstacles that universities have to overcome if they're going to be a real conduit of new technologies to get to industry.
So Ron, should I go to you, because although Seth sort of said that he thought that was the transition from academic to industry.
Yeah.
Is it not true also that biotech companies sometimes have that challenge when they're trying to take their technology into bigger pharmaceutical companies?
Well, I have a couple. So I heard you say a couple things. So one is the importance of translatability right now, so getting things into the clinic. And I think this is a theme that comes up a lot on AI and biopharma panels, because obviously many of the companies are quite young right now, and very few of us have actually taken the drug all the way through. So, at Recursion, we have two programs in the clinic, but one's only at phase 1, so we don't have that validation right now. The other thing I heard you mention is on reproducibility. And so when I think of reproducibility in machine learning applications specifically, the thing that comes to mind is the ability of these algorithms to pick up on substance in the data that might not be fundamental biology. And so, for example, we run experiments every week, and if we run the same experiment every week, there are experimental variations between those experiments. And if you just train a machine learning algorithm to try to predict the underlying biology, it's really easy for the machine learning algorithm to pick up the variation in the experiment and tell you these two things are the same, but they're not actually the same biologically. They're the same because they were run on the same day under the same conditions, and they have some, the staining looks the same. So when I think of reproducibility in the machine learning context, I think of the fact that when we're using isolated data sets that have been generated in one lab at one time and place, there's a lot of risk of being able to pick up on things that are not underlying biology. And so we need to be able to reproduce experiments across different contexts, across time, and then train models to be able to pick up on the fundamental biology underlying the signals that we're seeing.
Yeah, I'd like to add to that. Great points. We look for triangulation of data. So it's not just the reproducibility of any one type of data itself, but we're looking for triangulation from different types of data that all are pointing to the same, interpretation. And not really the interpretation, but actually lead to a testable hypothesis. And using this approach, we have actually now gone from hypothesis generation to 3 phase 2 programs in therapeutic areas as diverse as cancer, neuropsychiatry, and pain. So, and that's the beauty of using something like machine learning or artificial intelligence. You have to model it with different types of data sets so that you can overcome the natural variability that exists in data.
So John, I mean, you've been looking at this space for some time, both in your current role and also in previous roles. So, have you thought about where AI can again help in this this early stage research?
So I think the ideas of efficiency and reproducibility are very important, but they're kind of table stakes for the work that our clients are trying to focus on, which is insights and innovation. In big pharma, they've got massive amounts of data across many different, many decades of experimentation and studies. And what they're looking for is insights from cross-study analysis. So how can we look at all these experiments that we've done, improve our understanding of biology, and drive insights from this? And in order to do that, they're focusing on, well, what is it that we have to do to harmonize the data? And then what kind of intelligence and algorithmic approaches can we take to this cross-study analysis to derive these unique biological insights and new knowledge? And they're looking at the ability to do this as a differentiator, to create internal knowledge of cancer biology or immune cell biology that is unique to their team.
So Magdalena, you guys have developed a tool. What was the problem that you had observed that kind of instigated the idea in the first place?
So I think we all want to leapfrog to the moment where we have actually artificial intelligence, but at the moment we are training models to get us to that moment in time. And when we look at even not only academia, but also big pharma and biotechs, we still see a lot of information, like you said, being completely fragmented. Still quite a bit of information being in an analog paper format. And yet we speak to the AI tools and machine learning tools, and majority of the scientists in the industries are not even aware of these tools and don't know how to use them. So this is sort of what drove our development of our tools, that we want to create tools for scientists to enable them to teach the machine learning algorithms and to control them, because this is what is needed at the moment to refine these models to be able to actually offer insights and innovation later on. So our tool, we still think that reproducibility, it is sort of the foundation of research and development. And in order to do that, you really need to have not only capturing all the details that are happening in the lab, but also be able to connect the different data points and then offer insights to the scientists and people performing the work at the point of experiment. And a lot of times when you look at the tools today, they are very much disconnected from the work that's done in the lab. So this was what drove us, is to enable scientists to have access to information at the point of experiment and being able to document without an effort. So without having to go at the end of the day to their computer and type up what they have remembered they have done throughout the day, multitasking on many different experiments in a day. So I think we all want to get to that moment where we could provide the insights based on all the data we have. But right now, even in digital formats, we see completely hierarchical structure of folders and data that is not searchable and not discoverable. And what we want to do, what drove us, is how can we make this much more livable and interactive process.
Just to pile on that, frequently collaboration is increasingly important too. Collaboration across sites within one company, collaboration with an academic research organization, the NIH, a regulatory body, et cetera. And so the ability to be able to compare data across sites, to be able to analyze data across sites, requires information, metadata, about the experiment. What was the experimental method, the experimental approach, not just the actual data collected from the instrumentation. And so anything that can aid in collecting that data. Also, when you're doing cross-study analysis over time, you're comparing results from studies that were done this year versus studies that were done 3 years ago. You need that additional data, not just the experimental results that describe how the experiment was done, how the calibration was done, what the external and internal controls on the experiment were. And having all that data available is essential to really doing analysis that's going to be comparable.
Yeah, if I might add to this, I absolutely agree on collaboration. What we have seen, especially in global companies that have regional offices, that they're on a very foundational front. The scientists perform the work in their own native languages. And so you see one company that cannot access certain data created by another unit in another country because it is simply in a different language. This is also what we're trying to address with our tool. How can we enable NLP to translate research in real time?
So what you're talking about this, I guess sort of you're breaking down, or the siloed approach to some sort of research and development that takes place, whether it's within a company across different locations or where it's different partners attempting to collaborate with each other. That silo mentality, or the sort of the flow of data, what is the impact on the efficiency of the process? I mean, so how time consuming or time wasting is that? Does anyone have a sort of a thought of?
So, I think there's an inefficiency implied there, but much more importantly, there are barriers to knowledge creation, because in order to do interesting experiments are designed for the purpose of proving or disproving a particular hypothesis. And from the standpoint of one research program, the silos kind of work because you're doing the experiment to prove something, disprove something, etc. But when you look at an enterprise level, you're looking across programs, you're looking at the enterprise institutional knowledge about a certain area of biology, you need to be able to do analytics and generate insights and knowledge across all of this data. And that's where the silos create barriers, because it makes it very, very difficult to take that cross-enterprise, cross-program approach to your analysis and your knowledge generation.
Yeah, I'll just add that I think what we're seeing is that increasingly as we bring more technical sophistication into drug discovery, so being able to leverage machine learning for drug discovery, we need to have more cross-functional teams. And so it's not just biologists and chemists anymore, it's biologists, chemists, software engineers, data scientists, and everyone has to sort of work together and speak the same language in a way that they haven't before. So that's one thing. And the other piece is obviously the data, in that in order to actually be able to access the data coming out of the different parts of the organization, you have to be very thoughtful about how you're collecting it and how you're storing it, and ensure that it is accessible to the folks that are going to use it to train models and to be able to do the analytics.
And I'll add to that it's in a cross-functional team. It's very important that everyone is aligned to the same goal, because when you're looking at a difficult problem to handle, it's very important that everyone understands what the data is telling them, because the solution can come from different places. It need not always be pharmacology. It could be formulation. It could be in a clinical practice. It could be a companion diagnostic aligned with a drug. So no one group is going to be solely responsible for the success of a drug getting approved or being successful in the market. So when you're going through this very complex process, it's very important that all these groups understand what the data is telling them. They're all looking at the same data, but they all have different contributions to make to that data, right? And so they need to be able to communicate with each other in a way that is aligned to the same goal.
And how's that communication taken place up until now? In a pre AI world.
In a very inefficient manner of several meetings. I mean, this is why if you go to any big company, you have so many meetings, because we're trying to bring out the consensus as to what is the data, especially when a clinical trial doesn't work the way it's supposed to work. The post-mortem that happens, you're trying to understand what happened, right? Do we even understand what happened? And it's very easy to say, oh, that failed, throw the drug away and start something else. Well, you're going to repeat the mistake and you're going to fail again. So you really need to understand what is the data telling you, even when it's a failure, especially when it's a failure.
I think I just want to add to this, because this is similar in actually other industries where machine learning models are gaining traction, is that very often the transdisciplinary access and processing of that data in the model and feeding the model happens. It's a black box, and it's the same thing that happens in life science. Majority of these algorithms are trained and managed by engineers, and often are disconnected actually from the scientists that know the discipline very deeply. And I think that's something that I would like to see more of, having scientists much bigger access to the information that is fed into the machine learning algorithms.
I think there are a couple of issues here. One is an issue of language. People coming from different disciplines frequently have very different languages, and part of the initial meetings where you're starting to collaborate together is just normalizing on the terminology that you're using. A data scientist versus an experimentalist versus an IT person are going to have very different languages that they use, and they have to learn from each other and normalize on that. The other thing is the approach. They take very different approaches, even within the scientific community. If you have some scientists from a confirmatory function in the pharmaceutical company, interacting with scientists from an exploratory function in the pathway. We run into conversations all the time where the confirmatory scientists will say, you can't do that. And the exploratory scientists will say, we do this all the time. And so there are differences in methods, mentality, mental models, and approach that have to be brokered and understood as you're coming. And more, there's collaboration between the discovery scientists and the clinical scientists, because you're using clinical data or real-world data and combining it with the omics data to make these to do these analyses and discoveries. And these differences in mental model and language are barriers to getting to the substantive work of actually collaborating and coming up with the insights.
So one of the things, when it comes to sort of the recording of results, is that is sometimes important when it comes to your intellectual property and being awarded patents, etc. When it's written down, okay, whether it's committed to paper, etc., one can sort of see it's been dated. How do you sort of see are there going to be challenges around making those invention claims, using an artificial intelligence approach?
Well, we focus a lot in psychiatry, and we really admire the efforts of Psychogenics and a collaboration with Synovion, where they appear to have come up with a drug called SUN-856 entirely by artificial intelligence, using a cage in which the mice was photographed and all sorts of behavioral data was collected. So this appears to be the first anti-schizophrenia drug or antipsychotic that is not involving the D2 receptor. So I think that shows that the future of entirely new classes of molecules can come out of an artificial analysis approach. And I just don't think that this would have been possible by any other approach. So I think their intellectual property is extremely solid, although I guess to your point, the intellectual property is around the molecules that they've come up with, maybe to a larger extent than the way they found it. And by disclosure, I'm not involved in Synovion or Psychogenics. One of our directors was the founding CEO of Psychogenics. But I do think that a success like that, which I think looks to be probably one of the most important molecules that will come out in psychiatry for decades, I think that's a sign that everyone has to pay attention to this, and if you don't, you'll just be left out.
So I think there are at least two types of innovations. There's the innovation that's very deliberate and systematic, where you understand where you're headed and you take very deliberate steps that are explainable along the way. In that case, you can document as you go, and the regulatory bodies kind of like that approach because you can explain it. We like that as scientists. I think there's also the approach to innovation where someone has a brilliant flash, right? And you create an innovation where you have evidence that it works, but you're not sure why. And it may take many years and many different follow-on explorations to really explain why it works. But there's evidence that it works, and the evidence is reproducible. Those are important innovations as well, both in analytics and artificial intelligence. You may have an algorithm, you can't explain how it works, but it reproducibly gives the right answer. And biologically, you may find innovations in therapeutics that you can't really explain the underlying science by direct line of sight, but we know that it works. Our community needs to be more open to accepting in the breakthrough kind of brilliant flash innovations where we have evidence of the efficacy, but we don't necessarily know how to explain it.
I completely agree, and what we have seen also is that sometimes these flashes don't happen in the moments when you work on a specific target or specific experiment. Sometimes they may come up when you work on something that's completely different. And often you don't actually record it in a proper way. So this is also where we saw, and where a lot of scientists that we work with see value, is sometimes they have a flash of idea, and even though they work on something else, it's very easy for them to record that idea that is biometrically proven and has a specific timestamp. So later on, for IP purposes, these ideas can be tracked to the very first moment that they were discovered, make the traceability much more powerful at the end.
All right, so it's the Eureka moment that you're able to pick up.
Yes.
Yeah.
When I think about LabTwin specifically and the ability to document and interact with an AI agent while in the lab, I think of augmented intelligence kind of as three capabilities: understanding, reasoning, and learning. In the early phases of a technology like this, if it understands language, it can document what's going on when you speak to it verbally. When you think about now, as it's in the lab interacting with scientists, it will begin to be able to reason and make recommendations and understand what you're doing and give you hints or suggestions. And then it will get to the ability to learn, where it's learning from multiple scientists in different labs that may be doing similar work, or it may have learned about mistakes that shouldn't be repeated, or opportunities that should be seized upon under certain experimental conditions. As it develops the ability to learn, it will be able to interact with the scientist to interject and make recommendations based on that learning ability.
So Magdalena, what sort of proof of principle have you demonstrated so far on taking what John was saying there?
So we're early stage startup, so we're in the first phase. However, we've already have proven that many of our users, many scientists that we work with, say that now the work actually becomes discoverable, because something that was completely buried in a structured of folders that it could never be accessed easily, now the work becomes very iterative. So we actually now are, even though we are still more of we're just now in a moment where we're becoming a little bit more interactive and proactive, but we're still learning and still acquiring a lot of this information. And we've learned from our users that scientists are now thinking about the protocol that they annotated, that now they're running a 5th or 6th iteration. Now the system can tell them, well, you have done this 6 weeks before, and can sort of adjust the protocol much easier and much quicker than they have done before. But we're in a stage where we're actually acquiring the data now to learn.
And is that academic labs or is that industrial labs?
Both. So we started with academic labs, of course. That's where a lot of the innovation actually happens. And we're learning from that, and we are now also in biotech and pharma labs.
Okay, so you're sort of thinking about, okay, so there's some proof of principle, and we've had a kind of discussion around that there is AI is being used and has potential in this context. What I'd be interested, and I'm sure you'd be interested, Magdalena, as well, is what actually are still the challenges that exist to the adoption of such lab-based AI tools. So if you sort of think about that. I mean, John, do you want to kick off as what you sort of still see that the challenges that need to be resolved, and then maybe the other panelists could also give some thoughts?
Sure. I like to think from the user in to the technology rather than the technology out to the user. And the first obstacle is adoption, right? And in order for a scientist in a laboratory to interact with such a technology, it needs to provide some sort of utility to the scientist. What's the value to me? And so identifying that incremental roadmap of value so that they'll adopt it in the first place and then stay with it as it learns, because these technologies need to interact in order to get better. So they need the scientists to stay with it. So identifying that initial value for adoption and then the roadmap of incremental value drops is really important. The other thing is the data. We're finding with our work with clients that recording the data in systematically, all of the data that's required, and in a way that it can be conformed and harmonized is incredibly important. And so getting that right is important as well.
I think the very first question was how do you the challenge of translating early stage discovery and to the clinical thing, and this is where I think, and thinking about harmonization, the most important part of translating basic research or even clinical trial research into clinical practice is to be able to relate those findings in the lab and in clinical trials to the actual measures that have been taken in the real world. You can't do that if the language is different and the way things are being measured are different and the records are different. So the clinical trial records and the basic science lab data need to be related to real world data. So because there's going to be 100 times more people with 100 times more real world data than clinical trial data and lab data. And the real value comes when you're able to mine the real world data to see whether drugs are really effective, whether they're really safe. Can you actually learn from existing practice as how to design newer drugs? And you can't do this if the data can't be related to each other.
So Ron, I mean, you're sort of in a biotech company. When you sort of think about sort of the re-engineering that might take place, I mean, could bring in these sort of tools. How easy would it be to get the kind of the adoption that John thinks might still be a challenge?
Yeah, I mean, these are big questions. So I think right now we're at the stage where we're developing tools and we're testing them. So we're asking whether we can use machine learning in discrete applications in drug discovery and development. And we're seeing how these tools perform in those applications. But ultimately, that's if we just deploy tools that do very specific tasks, we'll get some efficiency. But that's not really what we're going after. What we're sort of trying to think about is, what does the world look like where we have access to computational tools, machine learning, for just about everything we're doing in drug discovery? How does that change our entire workflow from end to end? And that's the thing that's hard to imagine today, because we're just at the early stages of being able to collect the data, generate the data, and develop those tools. But it's something that as we bring more programs through the process using machine learning techniques, we'll start to refine as an industry.
So, and Seth, I mean, this sort of, in your company, again, this sort of this adoption.
Well, we can't wait for the right tools to be available to us. I look forward to these tools being available to us, but we spend a lot of time analyzing the data that we collect. We've done, in the past 5 years, 3 phase 3 studies and 2 phase 2 studies. The amount of data, the richness of that data is so important. And when you are analyzing a drug and understanding where it's going, particularly in psychiatry, which is extremely complicated, we look forward to going back and analyzing all of the text from all of the recorded conversations and all of the different measures and putting it together in hopefully entirely new ways. Because I think the future of an important part of the future of drug development in psychiatry is still going to be repurposing, and repurposing around clinical data, I think, is the richest way to be guided.
Okay, and as we're sort of now at the end of our time, I'll leave the last comment to you, Magdalena.
Well, adoption and behavior change, and actually culture change, is probably the biggest barrier. There's a reason that these environments haven't changed in the way we record data, even though we have all these amazing tools, haven't changed so much. And when you look at other industries, actually life science industry is quite behind when it comes to digitization. And the reason is that there's multiple stakeholders with these organizations that have different motivations. So if you think about the business stakeholders, the R&D heads, they want the data to be compliant, to be IP protected, and they have pressures to be more innovative and productive. So they adopt these tools that they've been pitched to and deploy them through our organization in hopes of having more data, better data. These tools very often are not designed and they engineered, but they're not designed with the user centricity in mind, which means the tools are incredibly actually difficult for most of the scientists to use in their day-to-day basis. So this is where I see the need. There's a reason that writing in your lab notebook is still prevalent, or in a piece of paper is still prevalent way of documenting, is because it is the easiest. And as human beings, we are all being a little bit lazy sometimes, and we always want to go for the easiest way to get to our goal. And when you think about that, that's missing in the tools that are provided to the scientists today. So what we hear actually from our users when we ask them, what if we took the LabTwin away today? What I love hearing, and we hear that a lot, is that they would miss being lazy. And I think this is actually a huge achievement, that now we have turned documenting into something that is it's not a hard and additional work for them anymore. And I think this is sort of what I would like to see us push. Can we create tools that are so seamless, so natural, and so fast that they create completely no barriers of capturing and accessing information, which will fuel innovation.
Okay, so when people want to sort of talk to you about this, how can they do that?
So I think if you grab the canvas bags, there should be my business card. If not, please come see us in our booth or look at labtwin.com. We actually just released our open APIs. We're not building another Yellen. We're not building another LIMS. We want to be a very lightweight platform that connects to existing systems. And for that, we are very much looking for partners, companies that we can connect to in terms of services and software and other tools. So if you're interested in speaking to us, we are very interested in speaking to you. Thank you.
Okay, so I'd like to thank the panels for their contributions, and also our colleagues at Bioprocessing for letting us use their theatre. And as I say, members of the LabTwin team are around, so they're available for additional conversations. So thank you very much.